be coming at the end of the linear structure, that will be the C-terminal amino acid. N refers to the, you can say nitrogen part, and C refers to the carboxylic, the carbon part. Then comes the secondary structure of protein. So this one, like for the example, you can say keratin, keratin of our hair, right? Our hair is made up of keratin protein. And then there is fibroin protein of the silk. So these two are the proteins and these are the secondary structures. Now see, when that linear structure, if I say linear structure, so it can be present in this way. So when this linear structure only, either it starts spoiling or it starts getting placed in the form of sheet. So that is how the primary structure changes into the secondary structure. How? Here, a protein thread does not exist throughout like a rigid rod. Sometimes it gets folded in the form of helix or it gets pleated. So when it gets folded, so we'll call it as alpha helix. And when it gets pleated, meaning it's present in the plate-like structure, we'll call it as beta pleated. So these two forms are the secondary structure forms of protein. Then comes the tertiary structure, the most important structure. Meaning, because of this particular structure, all of the enzymatic activities occur in our environment. Why? Because enzymes, they have a tertiary structure of a protein. So when long protein chains, they form a ball like this. Then that is the tertiary structure. They usually fold upon itself like a hollow woolen ball. And this is known as the tertiary structure. Now, this is a very important structure for the biological activities of proteins. That is for enzymes. Always remember the enzymes. You people know what enzymes are?
Yes, ma'am.
How do you explain enzymes?
They speed up a reaction.
Yes.
Biology.
Biocatalyst. Very good. So these enzymes, they have a tertiary structure because of which they have this function. Now the enzymes, they have a 3-degree protein structure. 3-degree means tertiary. Lastly, there is the quaternary structure of protein. So when the protein has more than one subunit, meaning when any one protein is a compound of several smaller units, we'll call it like a quaternary structure. For example, if we have hemoglobin, so hemoglobin has 2 alpha units and 2 beta units. So all in all, they have 4 subunits. So we say that hemoglobin is a quaternary structure. Clear, students?
Yes,
So
ma'am.
the structure of proteins are clear to you all?
Yes, ma'am.
Okay.
Yes.
Then the next biomolecule that we are going to discuss is the lipid molecule. Many a times we interchange the word lipid with fat. Okay. Now a lipid is made up of fatty acid and it is made up of glycerol alcohol. The oils, right? The coconut oil, the mustard oil, all the oils that we use, they are lipid in nature. Okay. Now remember that the lipids, they are not strictly a bio-macromolecule. We keep them under the bio-macromolecule category only. Why? Because their molecular weights are like, you can say intermediate and they are not soluble. Just like how bio-macromolecules are not soluble, same way lipids are also not soluble, because of which we keep them under bio-macromolecule situation, but they are not truly bio-macromolecule. Clear? So first you people note down till here, then we'll move forward. So you have to note down from primary structure, then
1 second.
secondary. Is there anyone who missed out? No one? Okay, now you people can note down. Wherever these rough figures are there, you note them down so that you'll remember. You can switch off your video and write down comfortably. See, Zainab, lipid is, it has both the kind of moiety in it. It is hydrophobic also, it's hydrophilic also. The tail is hydrophobic and the head is hydrophilic. Okay.
Yes, ma'am.
Done. Done, students?
Yes, ma'am.
Done. Everyone's done?
Yes, ma'am. Ma'am, could you explain the 3-degree protein structure?
See, 3-degree means tertiary. Okay. This is just how
Yes.
I've represented it with a number. So tertiary structure is only the 3-degree. When I say primary, it is 1 degree, secondary is 2 degree, and 3 degree is tertiary. Okay.
Yes, ma'am.
It's just the number numerical representation. The meaning is same. It's tertiary. Okay.
Yes, done.
Everyone's done.
Yes, ma'am. Done, ma'am. Done, ma'am.
All of you are done, students.
No, ma'am, 1 minute. Done, ma'am.
All right. So this is about the lipids that we are going to discuss. Now remember, this fatty acid, it has a carboxylic group. Okay. And these carboxylic group, they are responsible for attachment of the fatty acid components. Okay. So this one is attached to the R group. R group is again the variable group. Now when we talk about the lipids, so the lipids, they are water insoluble, and because of this very reason we call them as the biomacromolecule. That's just the only reason. Okay. Apart from that, their molecular weight do not exceed 800 daltons. Okay, their molecular weight do not exceed 800 Dalton. Now, normally, when we talked about all the biomacromolecules, their molecular weights were greater than 1000 or greater than 800 Dalton. But here it's not. But since these molecules are water insoluble, so we put them in the, you can say, biomacromolecule category. Hence, they are not strictly biomacromolecule. They are not strictly biomacromolecule. This is the unique thing about them. Now, remember when we talk about the lipids, okay, so they like the R group. The R group varies. In the case of lipids also, the R group varies a lot and it can be of variety of form. For example, this R group can be a methyl. And when I say methyl, it means that it will have just one carbon, right? Or it can go up to ethyl also, meaning it will have two carbons, C2H5, right? It can have two carbons also. So that is in the case of methyl. And even it can have higher number of carbon. For example, carbon 1 up till carbon 90. That is how the number of like molecules will be present in the R group. Now, same way, for example, if there are 16 carbon molecules, 16 carbon molecules, including the carboxyl carbon. So if we include that carboxyl carbon, if there are 16 molecules, so that is in the case of, like this is the example of palmitic acid. Palmitic acid is that lipid molecule that has 16 carbons in it. Same way, when we talk about the 20 carbons, okay, 20 carbons, again, including the carboxyl carbon. So the 20 carbon is arachidonic acid. Arachidonic acid. So this is like on the basis of the number of carbon molecules, including the carboxyl carbon are present. So we can call them as different names. Now remember, this fatty acid that we talked about, the fatty acids that we talked about, it can be of two types. There are two major types of fatty acid. The first one is saturated. Now, the meaning of saturated is that it will not have any double bond. So, no double bonds present. There are no double bonds present. And then there is unsaturated. So, here what will happen? Just the opposite. The double bonds will be present. So, this way, the fatty acid, they are of two types. Now, whenever the fatty acid combines with the glycerol, fatty acid plus glycerol. So there can be yet another so many different types. For example, after that, they can be monoglycerides. There can be diglyceride. And lastly, there also can be the triglyceride. Right? So you see how mono, di, and tri, these are the various kinds of the fatty acid when it gets combined with the glycerol. Clear, students?
Yes, ma'am.
Yes, ma'am.
Any doubts?
No, ma'am.
Okay. I told you that oils are the greatest examples of the lipid molecules. Now, what do you think is the difference between oil and a wax?
Ma'am, oil is unsaturated.
Okay, more like tell me in terms of its physical property. Tell me in terms of its physical property. See, if you have seen oil, then can you tell me, does it remain like a liquid throughout the year?
No, ma'am.
No.
No.
No, ma'am. During winter,
Yes,
it is hardened.
But if you think about the cooking oil, does it solidify?
No, ma'am.
No,
no.
they don't. So these
No.
oils, they have low melting points. They have low melting points. And have you people heard about suberin and cuticle?
Yes, ma'am.
What are they?
Yes. The waxy layer of plant.
The waxy layer of the that's present on top of the body of the plant. So what is the function of that?
Prevent water loss.
Yes, exactly. Okay, very good. All right. So if we look at the classification of lipids, we don't have to go in too much depth about it, but we should know that lipids, they are of three types, depending upon from where they originate. The first one is the simple lipids. The simple lipids. After the simple lipids comes the compound lipids. Compound, complex, or you can call it as conjugated. You understand the meaning of conjugation? In this case, it like normally bacterial conjugation is a different thing, but conjugation here refers that it is mixed with something. It is mixed with something. Then comes the last one, that is derived lipids. That comes from the lipids, meaning all those things that have been derived from the simple lipids. Now, the simple lipids further, they can be of three types. Let us see what are the types. The first one is true fat. The second one is the waxes. They come along the second category. And the third one is cutin and suberin, the thing that I asked you about right now. Then the conjugated or the compound lipids, they are further of four types actually. Now, you people, please pay attention and tell me how they are formed. If I talk about phospholipid, what do you think? What is this combination of?
Phosphate and lipid.
Exactly. Then comes
Phosphate.
yes, very good. Glycolipid, what about this?
Glycogen and lipid.
Glycose, no, it's carbohydrate and lipid, right? Then comes lipoproteins. What do you think? They are the combination of
Lipid. Proteins.
they are again fat molecules, the lipids along with the proteins. And lastly, there is chromolipids. These are certain pigments along with the lipids. So these are the different kinds, or you can say classification of lipid molecules. Clear? And have you people ever heard
Yes.
about cholesterol?
Yes, ma'am.
Yes. So that cholesterol, it comes here like sterols. One such example of sterol is cholesterol. So this is a derived, so the derived lipid. Any doubts up until now, students?
No, ma'am.
Okay. Now, when we talk about phospholipid over here, so we have like this one. It is the phosphorylated lipid, meaning the lipid that contains the phosphate. Example is lecithin. Do you know where this lecithin is present, students? These are found in cell membrane. Okay. These are found in cell membrane. So this was about the various kinds of lipids. And the other ones, they are not given in such a depth in your syllabus. So we'll not be discussing each one so in like a great depth. But for now, you people should know all of these things. Any doubts up until now, students?
No, ma'am.
So up until now, we have discussed the proteins, we have discussed the carbohydrates, lipids, right? All of these things are discussed. Now, the next thing that we have to discuss is the nucleic acid. The functions of nucleic acid, what are they, and how they are formed. Now, before that, do you people know what is a purine and a pyrimidine? Okay, we'll discuss that. Okay, don't worry. Even if you don't know, it's fine because you're studying it for the first time here. So we'll discuss the nucleic acid next. But the lipid is clear to you all students?
Yes, ma'am.
Okay, now you people
Yes,
can
ma'am.
note down. Okay, very good. The first 9 chapters are coming, Zanab.
Yes, ma'am.
Then name the chapters: living world, biological classification, then plant kingdom, animal kingdom,
Yes, ma'am.
then
Morphology of flowering
morphology,
plants,
hmm,
anatomy,
anatomy,
structural organization in animals,
okay, biomolecules.
cell, the unit of life, biomolecules.
So we have completed almost all of it, right? We just have to do structural organization, isn't it?
Yes, ma'am.
Okay, great. So relieved.
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am. What is written after no double bonds?
That is a plus sign along with NT, represents present. You can write present normally.
Yes, ma'am.
Done, ma'am.
Done, students?
Yes.
Done, ma'am. Done, ma'am. Just a minute, ma'am. Done, ma'am.
1 minute, ma'am.
Okay, students, take your time. It's all right.
Done, ma'am.
Done, ma'am. Done, ma'am.
Done, students?
Yes.
Done, ma'am.
Done, students?
Done, ma'am.
Okay. Okay, done, students? Now we'll be talking about nucleic acids. Now see, nucleic acid, they are the polymers of nucleotides. So they are the polymers of nucleotides. Now, first thing, if you want to learn about nucleic acid, so we must learn about nucleotides before that, right? And before that, like you have to learn about nucleoside also. So we'll see each one of those things, okay? So now when we talk about the nucleic acid, they are biomacromolecules. Since they are polymers, so you people understand it on your own that they are biomacromolecules. Now, after that, we'll discuss one more thing, that the examples. There are only and only two very important examples of nucleic acid, and that is RNA and DNA. Now, please tell me the full form of RNA.
Deoxyribonucleic acid.
RNA stands for
Ribonucleic acid.
ribonucleic acid, and DNA stands for
Deoxyribonucleic acid.
yes. Now, can anyone tell me what is the difference in between them? Hmm? What do you think is the difference? Both of them are nucleic acids, so what is the difference in between them?
Ma'am, in DNA, there's a sugar present called deoxyribose.
No, in DNA, the sugar is actually like the sugar does not have the OH group. So that is why that sugar becomes deoxyribose sugar. Wait, I'll tell you how. First of all, before we talk about the nucleic acid, we must know what a nucleotide is, because we saw here that the nucleic acid, they are the polymers of nucleotide, right? So when we talk about a nucleotide, always remember that it has three components. Nucleotides is equal to a phosphate. No, first we'll start with sugar. So pentose sugar. Pentose sugar means how many carbons will be there?
5.
Five carbons. Yes. Pentose sugar along with nitrogenous bases along with a phosphate group. When these three things combine, then that becomes a nucleotide. Okay, and these nucleotides only, they are the polymers of the monomers of the nucleic acid. Now, when we talk about the pentose sugar, the first moiety is the pentose sugar. Now, you people only told me that the pentose sugar has 5 carbons. So, 5 carbons. Now, there are two types of pentose sugar. There is ribose sugar And the next type of pentose sugar is deoxyribose sugar. Now I'll show you the structure. Okay, it's good if you remember. It's okay if you don't, but you can see the difference. See, this is a 5-carbon cycle. Okay, wait. Okay, so 1, 2, 3, 4, and then 1 comes out of here. So this is CH2OH. From here, there is OH on top, H on bottom. Here there is H, and then there is OH on the bottom. Then yet again, there is OH here and H here. So this structure, if we start to number the carbon, this is carbon number 1, carbon number 2, 3, 4, and this one is the 5th carbon. So this is the structure of the ribose sugar. Now when we draw the structure of deoxyribose sugar, so pay attention students. Now do you see the difference students? What is the difference in between these two sugars that I have drawn? See and tell me.
The second carbon, the
Yes, the second carbon of the deoxyribose sugar does not have any OH group present. And since the OH group is absent, we'll call it as a deoxy, meaning deoxy means no oxygen ribose sugar. At which place? So always remember that the deoxyribose sugar has no OH group at 2' carbon. Clear? This dash refers to prime. So I'll write it here so that you people remember. This is 2' carbon. Clear, students? So this is the pentose sugar. If it is RNA, then this sugar. If it is DNA, then this sugar. Does anyone have any doubts?
No, ma'am.
This sugar is clear, right?
Yes, ma'am.
Now the next thing that we talked about was nitrogenous bases. Do you people have any idea about it? See, nitrogenous bases, they are yet again of two types. Purines and pyrimidines. These are the two kinds of nitrogenous bases. Now, amongst the purines, there is adenine, that is represented by A, and then there is guanine, that is represented by G. Then comes pyrimidine. Pyrimidine, they are thymine, that is represented by T. Then there is uracil, that is represented by U. And then there is cytosine, that is represented by C. Now remember that this thymine is present only in DNA, whereas this thymine gets replaced by uracil only in RNA. So RNA does not have thymine and DNA does not have uracil. Clear, students?
Yes, ma'am.
yes,
All
sir.
right. Any doubts up until now?
No, ma'am.
Now see, the third thing that I discussed was a phosphate group. Phosphate group. Now this phosphate group comes from phosphoric acid. It comes from phosphoric acid. Who knows the formula for phosphoric acid? H3PO4. Okay. So the components of a nucleotide are clear to you all?
Yes, ma'am.
Yes, ma'am.
Okay. So if I say a nucleotide, then that means pentose sugar plus nitrogenous base plus phosphate group, isn't it?
Yes, ma'am.
Okay. Now there is one more thing called as a nucleoside. Nucleoside. Now see, whenever a pentose sugar is present with a nitrogenous base, a pentose sugar present with a nitrogenous base, meaning it is forming a bond, and that is what we call as a nucleoside. It is formed. Now there is the name of the bond. So the name of the bond is formed by glycosidic bond. Many a times it can also be called as glycosidic linkage. It's the same thing. Glycosidic bond and glycosidic link. Clear? Now, whenever this nucleoside gets attached with a phosphate group, then what do we have? Tell me. When the nucleoside, when I say nucleoside, then I mean pentose sugar along with nitrogenous base, right? So when the nucleoside combines with a phosphate group, what will it form?
Nucleotide.
Nucleotide. All of you are getting this thing? Is there anyone who's facing any difficulty? Tell me, students. Is there anyone who did not understand? Because I'll explain again. Everyone understood this?
Yes.
Okay, good. If you people understood it, then it's very nice. So whenever the nucleoside combines with the phosphate group, now the bond name is ester bond. So it becomes, or you can say it is called nucleotide. So now you people know the difference between nucleotide and nucleoside.
Yes, ma'am.
Okay, any doubts?
No, ma'am.
Yes. So this was about the nucleic acid. Now tell me, did you get the whole entire concept about it? Or is there anything that is troubling you? You can be frank. It's all right if you do not understand this in it at once. All of you have understood.
Yes. Yes, ma'am.
Okay, very
Yes.
good. Okay, there is one thing wherein we talk about how the particular structure, when it, you can say, forms a bond, becomes. I'll tell you about that. Just 1 second, I'll show you the picture only. For example, if I am saying that the nitrogenous base adenine is forming a nucleoside, can someone explain what do I mean by that? if I'm saying the nitrogenous base called as adenine is forming a bond as a nucleoside, what does it mean? Hmm?
It is forming a bond with sugar,
Yes,
pentose
it's
sugar.
forming. Correct. Meaning, see, if I say that the nitrogenous base is forming any kind of nitrogenous base, isn't it? So, for example, it can be different for all, right? Because we have so many different kinds of nitrogenous bases present. So, each of them can make different kinds of compounds, isn't it? Are you people getting what I'm trying to say?
Yes, ma'am.
Yes, ma'am.
Okay, very good. None of you are having any doubts. I'm so happy. I thought you people will get very confused and will have 10,000 doubts. So see this thing. I'll show you. Can you see the screen? See. Now this chart tells you that whenever the nitrogenous base combines with the this thing, the pentose sugar, right? For example, if we want to find out the nucleoside of adenine, right? Nucleoside of adenine means that adenine is combining with the pentose sugar, right? That's it. So that becomes adenosine. If adenosine is there, then that means the adenine has combined with just the pentose sugar. But when the adenine combines with the pentose sugar as well as the phosphate group, then that makes a nucleotide. So nucleotide of adenine is known as adenylic acid. This thing you people are getting?
Yes, ma'am.
Okay. Same way, same thing goes for the guanine. If the guanine is forming a bond with pentose sugar, then that is called as guanosine. Meaning guanosine is a nucleoside. But when I add phosphate to it, then that becomes guanylic acid. That is a nucleotide. Same comes for cytosine. When cytosine forms a bond with a pentose sugar, then the nucleoside is made. And this nucleoside is called as cytidine. And when this cytidine has phosphate group attached to it, then that becomes cytidyllic acid. These are the names of the compounds with individual nitrogenous bases. When thymine forms a group with the pentose sugar, so it is again a nucleoside. So the nucleoside of thymine will be called as thymidine, and the nucleotide of thymine will be called as thymidyllic acid. Same goes for uracil. The nucleoside of uracil will be called as uridine, and the nucleotide of uracil will be called as uridylic acid. Clear?
Yes,
Yes,
ma'am.
ma'am.
Any doubts?
No, ma'am.
All right. Now you people can note it down. Yes, the Annabelle explains better, Ryan.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes,
Done, ma'am.
Everyone's done? Did you people draw the structure?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Everyone's done?
Done now.
Everyone's done? Students?